So I'm now a sixth year. So I've done those first 2 years in medical school. It's my fourth year working in Melanie Ott's lab. I've spent these last 4 years mostly studying a different virus, HIV, which is has some similarities to COVID-19 in that there was an outbreak from animals and it's really had worldwide sustained spread in humans. And I have been in particular working on developing better diagnostics for HIV. So for the past 3 months only, I have been working on COVID-19 in the Ott lab at the Gladstone Institutes. And so some disclaimers, I'm not a doctor yet. The information is really rapidly changing every single day. There's new information, there's modifications to what we thought we knew about the virus. And so I will try my best to give you a sense of what the current accepted knowledge is now, subject to change. I view myself as a synthesizer and an interpreter of the facts and opinions from actual experts. There are really few true coronavirus experts in the world, people who really spent their whole lives studying this clade of viruses and people who really know and study pandemics. I am not one of them yet, and I would be skeptical of most people who claim to be or speak too definitively about this outbreak because I really think we don't know enough to speak in such a way. And of course, I do not speak officially on behalf of UCSF or the Gladstone Institutes. So for the past several months, I think we have all been deluged by headlines of varying, predicting varying catastrophes and economic collapse. As of just before this talk, I double checked, there are 407,485 cases worldwide. To give you a sense, that's just under half the population of all of San Francisco. And about 300,000 of those people are still sick. So meaning another 100,000 have either recovered or died. And so what I'm hoping today is to get away from kind of these sensationalistic headlines and get a good understanding of the information and facts that really matter that can inform our response. And so before we take a deep dive into coronaviruses, let's first take a step back. What is a virus really? One of my mentors in the lab when I first joined showed me this 1936 article from the New York Times where viruses were described as a new link bridging the gap between the animate and the inanimate, which offers evidence for the existence of a twilight zone between living and non-living matter inhabited by shadowy creatures. And I think really this is why I have long been fascinated by viruses, both philosophically, intellectually, and scientifically. They're really somewhere in between alive and not alive. And you can see that a tiny, tiny little virus has wreaked havoc like nothing else has managed to do. This is for me, I'm only 27 years old, but for me, this is the most drastic measures I have ever experienced living in the US for my entire life. Others have perhaps experienced things in a war akin to this, but I would say that it's very impressive that such a small particle that may or may not even be alive has wreaked so much havoc. So what is a virus really? In the most simple terms, a virus is simply a sack of proteins and genetic material. Viruses depend really on a host cell for raw materials, energy, and ability to multiply and spread. The first virus was discovered in 1892, the tobacco mosaic virus, where they found that when they took infected tobacco leaves and they took extracts from them and filtered them, which should have retained the bacteria, there was still something infectious there. And so they didn't quite realize what it was. The first time they actually called it a virus was in 1898. But they knew there was some particle that was infectious, and they knew that it was not a bacteria. And so because viruses depend on the host cell and they're not able to replicate alone, they are forced to be parasites. And what's fascinating is that they parasitize all biomolecular aspects of life. Bacteria get infected by viruses. Amoebas get infected by viruses. Protists, all animals, every form of life we have looked at, so far, if we look hard enough, we will find a virus that is able to infect them. So really, viruses are the smartest, inanimate organisms alive. And it's thought evolutionarily that they really date back to the very origin of cellular life. From the time there was first a single cell on the planet, a virus was there as well. And these two, over billions of years, have been evolving together. And what's most interesting about viruses that you may not believe now when you hear about the death rates for COVID is that most viruses are persistent and innocuous, not pathogenic. The goal of the virus is simply to replicate. The goal is not to kill the host. Killing the host is an often unfortunate and often rare byproduct of infection. And as I mentioned, because they date back to the very origin of cellular life, there has long been an evolutionary arms race between the virus and the host, each trying to evade each other, which we call the Red Queen hypothesis, where each organism must constantly adapt, evolve, and proliferate in order to survive, while pitted against another ever evolving organism. And then what I find extremely fascinating is that we often have a symbiotic relationship with viruses. And the placenta, actually, which is what differentiates us mammals from egg-laying animals, is that placenta, the protein that was essential for making the placenta, the syncytial cells, that is derived from a viral protein. So the placenta is only 160 million years old. Occasionally, some viruses actually insert themselves into our DNA genome. And it appears that a viral envelope protein was at some point captured during evolution into our genomes and was trained through evolution to operate in human biology and allowed us to form the placenta. So viruses are not all bad, is my point. So how do humans get new viruses? We're constantly inundated by exposure to viruses. And really, for the most part, we don't think of viruses as super harmful. Everyone gets a cold or a sore throat occasionally, and it usually doesn't cause much trouble. Now, viruses are constantly mutating. And so new viruses could emerge just by mutations in the currently circulating viruses. And so unless the changes are major, usually our immune system, through prior exposure and training and this evolutionary arms race, is able to handle it. But sometimes completely new viruses appear that humankind has never seen before. So let's take Ebola as an example. Here you can see that what we call the virus reservoir, or an animal where this virus can replicate but usually doesn't cause disease, in Ebola is thought to be bats. Sometimes the bats might have feces that fall on the ground, or plants or water might be contaminated by the bat's saliva or feces, and a wild animal such as monkeys might eat that fruit that's contaminated and become infected with this virus. When often it's thought that the first crossover from Ebola to humans occurred when a bushmeat hunter potentially was butchering a monkey and may have cut his hand, and the virus was previously infecting the monkey and now infected the human. And then sometimes, not always, the virus can actually be able to facilitate human-to-human infection, where we get sustained transmission. Oftentimes we'll get a single human be infected, but they might be the dead end host. The virus is not able to adapt to the human and not able to infect new people. But by pure luck and by increasing interaction with wildlife, we do increase the chance that a virus could potentially get lucky, be able to replicate in the human, and we would get sustained human-to-human transition. Now, I've been hearing a lot of people saying, we should ban these live animal markets in China, and sometimes bordering on kind of racist takes on, why we have this virus. But let's not forget, in 2009, the H1N1 swine flu that originated on this side of the world. And we come into contact with livestock all the time. How many of us have gone to or taken our children to petting zoos? An infectious disease doctor would never allow their child to pet animals at a petting zoo because we as animals are closely related. And so that means that a virus that could infect a pig could also have the potential to infect a human. And so the H1N1 swine flu was actually a really interesting virus, where there was a human flu virus, an avian flu virus, and then also a swine flu virus. And these three viruses inside one pig that happened to get infected by pure chance with all three, there was some different combinations between the flu that created a brand new strain of influenza that we as a human population had never seen. That, through having pig farms, led to pig to human transmission and then sustained transmission in humans, leading to the 2009 pandemic. So now we know a little bit about viruses. We know a bit about zoonotic transmission, which is what we call this jump from species to species of viruses. Let's get into what coronaviruses are. So on the left, we have a electron microscopy image of viral particles, and they're named corona, or which is Latin for crown or halo, for this kind of crown-like appearance or halo-like appearance around the viral particles. And some of the kind of more important features of the virus to know, if you were to cut the virus in half and look on the inside, it has a genome. So we have a genome made of DNA that we inherit from our parents, one copy each. And the viral genome is actually made of RNA, which is different than our genome. And this RNA, like I said, it's a stack of protein and genomic material. So it's RNA and a protein inside the virus, and it's surrounded by this lipid bilayer and some spike proteins, is what they call them. Now, two really important things. So number one, because this virus is encapsulated with this lipid bilayer, that means that soap is actually really effective against this virus. That's why everybody's been begging you to wash your hands. Soap is able to disrupt this lipid bilayer and disrupt the virion so that it's no longer able to infect you. And so by washing your hands well, you're destroying the virus and you're prying the virus apart. The other thing that's really important to know about is the spike glycoprotein. This is what the virus actually uses to attach to the human cells and infect us. We know a lot about this spike glycoprotein because the spike glycoprotein in SARS back in the early 2000s was very similar. And so very quickly, as soon as we had the genome for this novel virus, we were able to figure out by genetic analysis how similar the two viruses were and make a prediction that it would be very likely that this SARS-2 or COVID-19 can infect the same cells as the original SARS. And this is called the ACE2 receptor. And so this is expressed on lung cells. So this is why you get respiratory infection. It's expressed on gut cells, which is why sometimes diarrhea is reported as a symptom, although rare. And I'll get into the clinical manifestations. But the other thing that I'll say is that so far we've discovered 7 human coronaviruses. 4 cause the common cold. We have probably all been infected with them. And then the other 3 is that SARS in the early 2000s, MERS or Middle East Respiratory Syndrome, and now SARS-CoV-2. The first coronavirus, we've known about these viruses since 1937. And we know that they can infect cattle, pigs, cats, ferrets, turkeys, mice, dogs, rats. And so really these are ubiquitous in our environment. And so we should expect that this is not the last coronavirus that we will be exposed to. So where did COVID-19 in particular come from? So COVID-19 is the disease name. It stands for coronavirus 2019. So this is the disease. It's caused by the virus called SARS-CoV-2. So this is the disease, this is the virus name. Just before the new year, China reported this cluster of pneumonia of unknown etiology. And the first cluster of patients emerged in Wuhan, Hubei province in China. And it seemed to be associated with people who either frequented or worked at a seafood or live animal market. However, I will say that there's still been some debate as to whether the virus actually originated from that market or somebody got infected who worked there elsewhere and then came to the market and spread it there. It's now been declared a pandemic. What is a pandemic? A pandemic is a global outbreak of disease. And by early on in China, the first few patients, they sequenced the genome of the virus. This allows us to compare it to all known sequences in the world. And we found that there's great sequence similarity to some bat viruses, that people had gone out, they had gathered fluids from different bats and sequenced the different viruses that were in there. Like I mentioned, Ebola is also thought to reside in bats. And so bats really contain lots and lots of different viruses that don't make them sick, but could make us sick. And so it's thought that bats are likely the original reservoir, but that there may have been an intermediate host. Some have suggested the Malayan pangolin, which is this kind of interesting, scaly, sloth-looking creature. You can see here that they sometimes illegally use in many parts of the world. And then there was some type of final zoonotic transfer to humans, is what kind of these genomic investigators have figured out so far. Um, and then what I will say is that we haven't found an animal coronavirus yet that is exactly close enough similar to this virus where we can say that it is the direct progenitor of this virus. And that's really because the diversity of coronaviruses and viruses in general in bats and in other species is massively undersampled. So what we really need to be doing is hunting for these viruses actively in animals and at interfaces between humans and wildlife to get an idea of what the next pandemic could be. And I'll reiterate this later in the talk. And so the idea is that if this virus is pre-adapted in another animal species, there could be future reemergence events. You may see that Ebola every few years, it seems to disappear and then it reemerges in the human population. So what is the clinical manifestation of disease? And you know, there's a lot that's changing about this virus, but I will just give you guys some clinical pearls that I have heard about both at UCSF and University of Washington, where the first cases in the US were sequenced by actually a graduate of the MD PhD program at UCSF, who I overlapped with. And so the common triad is a fever, not necessarily high, sometimes it can be low, cough, and shortness of breath. And so fever is seen in about over 75% of hospitalized cases at some point, but almost 50% are afebrile upon admission. Cough, we see in about 60 to 80% of patients. Shortness of breath is in 20 to 50% of patients. So you can see none of these are really the definitive factor for diagnosing someone with coronavirus just based off clinical symptoms. And so a lot of times they had been relying on prior travel history, but now there has been so much sustained transmission and asymptomatic transmission in the US that it's hard to really just use travel history as a good indicator for the odds that they might have coronavirus or not. Other symptoms, many people report myalgias, which are body or muscle aches. Between 10 to 50% will report that. There are upper respiratory symptoms such as headaches, sore throat, runny nose, but they're not as common. They're seen about less than 15% of hospitalized patients. And then there are, like I said, there's receptor for the virus is expressed on gut or gastrointestinal cells. And so we do sometimes see gastrointestinal symptoms, diarrhea in less than 25%, nausea and vomiting in less than 10% of hospitalized patients. And you know, the most concerning aspect of this is that there's often a progression to pneumonia. So here you can see on the left, these are CT scans. On the right are chest x-rays. So this chest x-ray here, this is would be a non-severe case where you can see the black area is really outlining the lungs. The lungs should be filled with air, so it should mostly look black on an x-ray. Here in the severe case, you can see that because the lungs are filled with fluid and virus and pus and dead cells, which is what pneumonia is, they turn this kind of opaque white color. And so this would be a very severe case of pneumonia. And the classic pathognomonic finding on the CT scan is what we call ground glass opacities, where taking basically cross sections of the lung, this is the heart here, these are the two lung fields, they should look black and relatively clear, but you can see that they're occluded and they kind of look like if you had cut up glass and sprinkled it in there. And so really the virus is very smart. As the virus infects a lung cell, it will start multiplying in the cell. The virus will want to burst out and infect neighboring cells. And so it'll crawl progressively down your lungs and infect different cells in your lungs. And then when your mucous membranes become inflamed, when your lung sacs or alveoli become inflamed, you start to have trouble carrying out the function of supplying oxygen to the blood that circulates through the lungs. And so that's why you get this shortness of breath, because your lungs have filled with this pus, this fluid, these dead cells. You start to have difficulty getting good transfer of oxygen from your breath to the actual lung cells and into the blood. And so this is why a lot of people end up needing to go on a ventilator. This is why even young people sometimes need to get supplemental oxygen in the emergency room or in the hospital. So who is especially susceptible? Well, as it turns out, age is a major factor, age 65 or older. In the US, 8 out of 10 of the deaths have been in adults above the age of 65. Up to 27% of adults above the age of 85 or older have died. That's pretty bad, is what I would say, compared to most other viruses. And among the age group 65 to 84, between 4 to 11% of infected adults have died. Comorbidities, so things that can make you have a more severe course of disease, include diabetes, any chronic lung disease, cardiovascular disease. In China, the odds of progression to severe disease was 14 times higher in smokers. So if you've ever been looking for an excuse to quit smoking, I would say now is the time. And then those who are immunocompromised. So people who get cancer treatment, they're an organ transplant recipient. Some people have autoimmune diseases where they need to calm down their immune system and take different drugs. Those people are especially immunocompromised, even if they are not above the age of 65, and especially if they're above the age of 65. And then nursing homes, they tend to be bad sites of outbreaks in general, but because there are often comorbidities, because the age is high in nursing homes, that's really just, if coronavirus ends up in a nursing home, that's someplace that we're going to be really concerned about, and there's going to be a high level of morbidity and mortality. And on the right, you can see a graphic created by BBC, where this is based off of the Chinese data, but you can see it mirrors kind of the preliminary data that we have out of the US, where the age, really, if you're above the age of 65, you're at quite high risk of death. These health conditions really increase your chances of death. And there doesn't seem to be too much of a predisposition for male or female. It's thought that in China, maybe more of the males were smokers. And so potentially they might see an increase in deaths in males. So why is this virus so particularly bad? Well, there's a few reasons. Like I said, the virus is new. It's novel coronavirus, is what it was originally named before we came up with the name COVID-19. And so our immune system has never really seen this. And so unless you have been previously infected with SARS, which even then we are unsure about, it seems that our immune system, this is really new to our immune system. And so we don't have this evolutionary history of seeing this coronavirus to be able to mount a good immune response to it. In this chart that is from the New York Times, but there have been many versions of it, they're graphing on the y-axis the fatality scale on a log scale. So it goes from 100% to 0. And then on the x-axis, you have the average number of people infected by each sick person, which is called the R0. And so I will go into more depth about that on the next slide. But here in the red square, you can see we have about an estimate of what this new coronavirus has. Most estimates seem to be putting the fatality rate below 3%, although in Italy, it's been much higher due to their higher elderly population or at-risk population, and the number of transmissions from each sick person between 2 and 4. So you can see, if we were to compare this severity to other known viruses, the common cold, the 2009 flu that I spoke about before, those are much less fatal and much less transmissible than this new virus, potentially. In fact, this is less fatal and less transmissible potentially than even polio. MERS is extremely fatal. Nearly 30% of people infected with MERS die. But with MERS, you generally have to come into contact with a camel, and human-to-human transmission has not been as much of an issue and as widespread as we've seen with this new coronavirus. Ebola tends to kill most of the people it infects, but because it's so quick and so hot, you don't get to transmit it to as many people. But the other thing that is really interesting about this virus is that there seems to be asymptomatic spread. It could take between 2 to 14 days where you're infected with the virus. You could infect new people, but you have no symptoms and you have no idea you're infected. And potentially even, you may never develop symptoms. You may clear the virus, but you may have infected several people. Now, I mentioned that number R0 before, just to give you an idea again of other viruses. So R0 estimates really how many other people one sick person is likely to infect. So for the new coronavirus, it looks like 2 to 3 people, although again, this is preliminary. Zika, between 3 and 6. Measles is very infectious, but not that deadly. Ebola, you're so sick so quickly, you don't even get a chance to infect other people. And you can see the seasonal flu, you're likely to only spread it to one person. Now, again, why is this virus so bad? So I've mentioned that it's completely novel, so our immune system doesn't really know how to handle it. There's asymptomatic spread, but I think that this chart really makes us understand why testing is so important to getting ahead of this. So here in the red charts, they have the cases in Korea. In the green, they have the cases in Italy. In Korea, they really immediately took charge at the start of this pandemic and started testing people. They had drive-through testing. They were testing more people in a single day than the United States was able to test total as a country over a month. I mean, their testing capacity was really admirable and remarkable. And so because they did such widespread testing, they weren't limiting the test to just very sick people like we see here, where we don't have enough tests. There, they were able to test everybody. And so what you saw in terms of the age distribution is that when you compared it to the actual age distribution of the total population, it really followed it. And not only that, but you see in the age 20 to 29, nearly 30% of cases were in that age group. But likely most of those people were asymptomatic. However, in Italy, number one, they have a very large elderly population, much higher than Korea, which has a much younger population. And so you really see most of the cases are in, above the age of 50 here. I would say nearly 80% of the cases are above the age of 50. There's been a lot of fatality because this age group is particularly susceptible to death. And then on top of that, they don't have testing to test everybody. They don't have enough tests to test those people who are young and asymptomatic so that we could potentially isolate them and prevent them from testing other people. They were forced to go on a nationwide shutdown. Here, we also don't have enough tests to test everybody. I have not been tested. Most people I know who are my age have not been tested. Even those who are symptomatic are often not able to access tests unless they need to be hospitalized. And so we really don't know. And because, New York, many states have been resistant or even threatening to lift their lockdown orders, these asymptomatic people will go back out and they will continue to infect people. And then the other thing that I would mention is that, although young people tend not to die, when we do get infected and when we do get symptomatic, we still take up hospital beds. And so the other issue in the United States is that we don't have enough beds, we don't have enough ventilators, and we don't have enough protective equipment. And so if young people, even if they're not going to die, but do need supportive oxygen, they may need the help of a ventilator briefly, or they may need to be hospitalized to monitor their pneumonia. They're taking up beds that should otherwise be used by really sick elderly people. And so in the United States, really the problem has been compounded over and over again. Instead of being managed the way South Korea did, by early and rapid testing of lots and lots of people so that we can identify the infected and isolate them and let everybody else continue on with their lives. So I've said this basically so far, but the US response has been, in a single word, abysmal. And so in part, it was the testing failures and mismanagement. So originally the CDC, the Centers for Disease Control, decided to create a test themselves. As it turns out, their test is not very good. I have tried it myself in the lab. I found it to be neither sensitive nor specific. And I've moved on to testing kits from other countries. Not only that, but initially they didn't even realize how bad the test was because they had manufacturing issues where their controls were not good controls. So it took a long time. And because they had centralized it to just the CDC giving out the tests and running the tests, essentially other labs were limited from running their own tests until a lab at the University of Washington basically went rogue. They couldn't get access to the test, and they just started sequencing the entire genomes of patients. And that's how they discovered that they had an outbreak in Washington State. And now they are becoming much more, accepting of allowing each place to develop their own tests. Now UCSF has created their own tests. Stanford has created their own tests. But you can imagine if we had both competent and organized national response, every hospital and university wouldn't have to de novo create their own test. We could have one that was both effective, unlike the CDC one, and widely manufactured and distributed so that we could get our testing capacity up. The second issue is the lack of ICU beds, ventilators, protective equipment. Doctors are basically not going to have masks or gloves to protect themselves from patients, partially because despite the fact that China really, I would say, bought us time to get these things ready. Nobody responded to the call. Nobody saw the warning signs. And so we didn't start increasing our manufacturing. We didn't start stockpiling these things. And so in San Francisco, at least, the UCSF medical students, who some of their medical rotations have been canceled in light of this, they have been standing on a street corner and collecting from ordinary citizens any gloves, any masks, any equipment or protective equipment that they have that they could give to the doctors, because within 2 weeks we're going to be out, and it doesn't look like we're getting much more anytime soon. Although I did see today that potentially Donald Trump has authorized the government to take their war powers and to start producing masks, which we also need them to start producing the ventilators. So really, the United States failures have been on many levels, and have continued to compound each other. So let's take a look at how bad it really is and where we could have been. So let's take China, for example. These curves, the way to interpret them is that on the x-axis is the number of days since the 10th death, and on the y-axis here is the number of deaths. China, pretty early on, they have faced a lot of deaths, but you can see that they're not really getting new deaths at this point. Within a month, they were able to start containing it, and actually for the last several days, they have reported no new cases and no new deaths. South Korea instituted an early and large-scale testing, and they really took their public health departments and they started tracing the contacts of every known infected person so that they could tell them to isolate. They really put their public health departments to use and to work. And so they got the outbreak under control very quickly. In these dotted lines, these are telling you how the rate of the deaths. So here in this, if a curve follows this line, the deaths are doubling every day. If the curve follows this line, the deaths are doubling every 2 days. Here it's every week. And you can see South Korea, out of all the countries, because of these interventions, and keep in mind they're a democracy, so it doesn't require an authoritarian government to really stop a virus. But they really I think, are the model for how to handle a pandemic. On the other hand, we have the United States, which is, I think, is right here. You can see that our curve is still going straight up and doesn't seem to be flattening anytime soon. Spain and Italy have really had very scary, I would say, outbreaks there. In Italy, because they have such a large elderly population, the death rate has been just astounding. They haven't even had as many cases as China, but they already have twice as many deaths as China. And Spain also, it looks to be pretty uncontrolled growth right now in terms of the death rate. And you can see the stars about when the countries decide to lock down. In the United States, unlike the UK, France, Spain, Italy, has not had a nationwide shutdown. It started with, our counties here in the Bay, now California, and a few other states. Now, let's take a closer look. So one of the places that concerns me the most is actually New York State. You can see they are basically going straight up. And if they follow this current curve, their deaths are going to be doubling every single day. And New York currently has the most cases and the most deaths of any place in the United States, despite the fact that the virus first arrived in the US and Washington. California has been doing quite well, I would say. And I think this is in part due to our social distancing. But like I said, it takes 2 to 14 days for those people who are potentially infected to show symptoms. So I would say it would take another week or two before we really know if this lockdown has had an effect. But I would say that in California, it's promising, and it looks like we're starting to get things under control, unlike New York. Madrid has been very hard hit, and Lombardia in Italy has really been devastated by this. So is there a vaccine? Is there a treatment? So vaccines, people really mobilized on the research side as soon as they found out about this virus. And so there are several underway actually in vaccines and trials. But the problem with vaccines is, it takes a long time to validate it. And it takes a long time to get, enough vaccines where we can actually vaccinate everybody. So, we have lots of vaccines underway, but it'll take about 12 to 18 months, assuming all goes well and the vaccines are actually effective. So in that year to year and a half, what can we do in the intermediary? Well, as it turns out that we can often repurpose old drugs to new purposes. And this has been done many times in the medical field. So to give you kind of a brief overview of the drugs that people are using at UCSF, at other institutions, or the World Health Organization announced kind of the biggest randomized controlled trial of several of these drugs the other day. So the first one is called remdesivir, and it's produced by Gilead. I really admire Gilead as an HIV researcher. They, a lot of companies will not produce drugs that will treat infectious diseases because it's thought that infectious diseases only affect the poor, and so they're not profitable the way a statin drug for your heart might be, or a cholesterol drug might be. And so Gilead has really been a leader in infectious diseases. They have a great hepatitis C treatment. They have a drug for HIV, and they created this drug originally for Ebola. And in the lab, some coronavirus researchers many years ago showed that it could potentially be effective against SARS or MERS. And so this, I think, is a really promising drug. It still remains to be seen if it'll be effective against this particular coronavirus and if it will actually be efficacious in patients and reducing the mortality. But there are studies underway all over the world and at UCSF. Other drugs that you may have heard about in the news recently is chloroquine and hydroxychloroquine. They were originally developed for malaria prophylaxis, and also they're used for treatment of autoimmune diseases such as lupus. Our president, Donald Trump, recently announced it as the cure, which there's one preliminary study that was very poorly controlled, is my opinion, that does not definitively show if it has any positive or negative effects. And in fact, these drugs are quite toxic. And so, somebody heard Trump say that this is the cure, this is how you prevent coronavirus. Yesterday, he and his wife took a derivative of chloroquine that's used in aquariums. Actually, he and his wife took a bunch. He unfortunately passed away, and his wife is now in the ICU. So again, facts, information, these things matter. And touting things as cures when we don't have any evidence and we don't have any control is extremely dangerous and costs people lives. So the other drugs that we're looking at is ritonavir and lopinavir, which are drugs that are originally used for HIV and target that viral protease. But because there are similarities between viruses, you could potentially use it against this coronavirus. And then it's also thought, I mentioned that pneumonia is really an inflammation of the lungs where you have fluid filling there, you have dead cells, debris from these viruses bursting these cells open. And so potentially some of the death is because there's so much immune activation that you go into acute respiratory distress. And so if we could calm down the immune response potentially without targeting the virus, perhaps patients would recover better. And so IL-6 is a cytokine or a molecule that our cells release to stimulate an immune response. But if we can block this, and this has previously been used for autoimmune diseases such as rheumatoid arthritis, maybe we could reduce immune activation and inflammation so that the body doesn't get overactivated. So these are things that people are actively trying in the clinic. And I hope very soon we'll have some good controlled data to tell us, are these efficacious or not? What are researchers doing about it? So I'll just briefly, before I end, go through kind of two big things that us at the Gladstone Institutes have been working on and at UCSF. So the first thing, you may have seen a couple articles in the New York Times this last week. So this is from the QBI Coronavirus Research Group. And basically what they do is they take each individual protein that the virus takes. So the viral proteins are in the center here: E protein, M protein, N protein, non-structural protein 1. And they are able to study what human proteins these viral proteins interact with. Like I said, viruses are parasites. They require our host human proteins to replicate and to operate inside the cell. So we know from lots of our other research that viruses interact intimately with the human proteins. And so if we could identify what human proteins are essential or necessary for the virus to replicate, and we already have drugs against those human proteins, we don't need to create from scratch a new drug against coronavirus. We can just briefly block that human protein with a drug, and then we could prevent the virus from replicating and causing disease. So one thing that's really interesting to me, because our lab has studied it in the context of HIV, is this protein called BRD2. And it's close relative BRD4 that interacts with the viral envelope. We actually already have drugs in it. That's also an important driver in cancer. And so we have several drugs that have been used in human trials to block this protein. And so this study was just published yesterday online for all international researchers to be able to look at this data and start testing drugs. And so we're really trying to make our data open and really trying to collaborate as much as we can across the globe to as quickly figure out what drugs might be effective in the clinic, what new drugs in addition to the one that I mentioned before. So this is just a taste of, this is really led by Nevin Krogan's lab at UCSF and the Gladstone Institutes. And I think there were like 10, 15 groups overall involved in this Herculean effort of getting this done in a month, where this would usually take, years. My own research, I mentioned that I've been working for a while with HIV diagnostics. The nice thing about the diagnostic system that I've been working on, which the goal is to have it as a self-test or something you could use at home to detect HIV, originally in my case, is that it's easily adaptable to new viruses. So when we realized what a testing shortage there was, what a long testing turnaround there was, sometimes it can take a week for you to get your results. We decided that we should really adapt this kind of mobile phone-based testing that could be done either in a clinic, at a border, an airport, or potentially even your own home to the coronavirus. So I've been working for the last couple months trying to adapt this. The idea would be that usually it's diagnosed by a nasal swab. This thing goes pretty far back in your nose, I will say, but it's totally doable to swab yourself or have someone swab you. I've done it. And you would take some nose tissue that's near the back of your nose. And if there's any cells that are infected with the coronavirus, you would also isolate those viruses. You would stick this swab into a device. We would cut open the viruses so that we just have that RNA genome. And we have this new technology called CRISPR-Cas13 that is able to really detect very specific pieces of RNA. So we programmed these for the coronavirus. And if they find any coronavirus RNA, which is the genome of the virus, they will also cut a reporter that will fluoresce that you could use your own iPhone to hook onto this device and detect if there's fluorescence or not. So within about 30 minutes, you could have pretty accurate quantitation of whether you're positive or negative for this coronavirus. So finally, what should you be doing about the coronavirus? So social distancing, learn to love it and flatten the curve. The idea of the curve that you have probably seen many times is that without protective measures, we will get a massive rise in cases that will overwhelm our healthcare system capacity. We don't have enough masks, we don't have enough beds, we don't have enough ventilators. If we all get sick at once, people are gonna die just because we have no place to put them. On top of that, we will infect the healthcare workers who will no longer be able to take care of us. And then people who are not infected but have, continue to have heart attacks, strokes, other things that send them to the hospital, they will also die for no reason. However, if we practice social distancing, if we go on lockdown, if we socially isolate, because we don't have enough tests to go around right now, we can flatten the curve, meaning it would take longer for people to get infected. It would be slower though. So, maybe there would be 50 cases here, maybe 100 at the peak, but we would never overwhelm the healthcare system capacity, which would reduce our death rate because we can actually take care of everybody. Like I said, please wash your hands. Soap is extremely effective. Now you know the scientific reason why, so there's no excuses. Use common sense measures. When you do go out to the grocery store, really keep your distance from other people. Stand on those lines 6 feet away from the person in front of you in the checkout line. And then if you have any personal protective equipment at home that you don't need, such as masks or gloves, please donate them to your local hospital because they are in desperate need. And they are the ones who will be coming into contact with the virus, and you at home hopefully will not. And so, what about the future of humanity? I did say, if we could cure this or we could get rid of it from humans, but if we keep coming into contact with wildlife, we could potentially get re-exposed to this exact same virus or other viruses, right? And, actually people have been predicting this for a long time. Someone who I would consider a true coronavirus expert, Ralph Barrick, who's at the University of North Carolina, in both 2015 and 2016, warned in prominent papers in scientific journals that there is a SARS-like cluster of circulating bat coronaviruses that show a high potential for human emergence. And so if we continue to do basic science research where we're really going out into the field, we're collecting samples, and we're understanding what is the viral diversity out there, because again, we had never seen this virus before until it showed up in a human. We could start to get an idea of what things should we preemptively start making vaccines for, what should we be studying in the lab, so that the next pandemic, we're not caught, flat-footed. And, I would say that there is no escaping viruses. Viruses can be our friends. They can be our symbiotic relationships with us. And, even if we go to Mars and set up a new colony there once we've destroyed the Earth, there are viruses on your skin. There are viruses in your gut, in your throat, in your nose that we will take with us. And so there is no escaping them. But through climate change and globalization, climate change by changing the habitats of wildlife that will force them to come into contact or restrict their habitats, globalization where we encroach into animal habitats, those will all lead to more interactions with wildlife that potentially harbor novel infectious diseases that we have no predetermined immune response to. So really, this should not let us forget how important it is to mitigate climate change and how important it is to be mindful of our globalization efforts. And, the more we fly, the more we travel across the globe, the greater the risk is that we could spread these new viruses, which is exactly what happened here and what always the fear is with Ebola, that somebody might bring it to the US and there would be local spread here. Again, please lobby your congressman, your senator to fund basic science and biomedical research in infectious diseases in public health departments. Public health is frequently underfunded. Infectious diseases doctors actually take a pay cut from just being a regular internal medicine doctor to get 2 years of a specialized additional training. Like I said, people think that infectious diseases only affect the poor, and so we don't see a financial benefit into pouring money into it. So we really need to lobby our government to take infectious diseases seriously, because you can see what a tiny little virus has done to the global economy. And then finally, international cooperation and heeding warnings is key to future pandemics. I would say that China really bought us time and really did the world a service. We did not heed their warnings. We did not take that month that they gave us an advance warning to get things together in the United States. We were ready for the response. And this is really why, if we had widespread testing, we would not be all in social isolation and lockdown. This is really a failure of government and a failure of competence. So with that, you may have or may have not gotten my initial reference to my favorite movie, Dr. Strangelove, or How I Learned to Stop Worrying and Love the Bomb. But thank you very much for your attention, and I'm happy to take any questions.
Thank you so much for that educational update to us all. Such a hot topic everybody's wondering about. How much time do you have for questions and answers, just out of curiosity?
I have about probably like 10, 15 minutes.
Okay, so great. I do have one.
Can you unshare your screen, and that way we can see who has questions? Thank you.
So one first question for you. I saw in the news yesterday that the Diamond Princess reported traces of coronavirus inside the cabins over 2 weeks after they had pulled everybody out. So how long does this virus stick around?
Yeah, so there was a study out of the NIH a few weeks ago that caused some traction, where in a laboratory setting, they had aerosolized the virus into the air. They had put the virus on different types of different types of materials to see how long it would last. Those studies are difficult to interpret, at least for me, because the laboratory setting is so different than real world, right? So aerosols, typically when we cough, we're not creating aerosols, we're creating droplets. So droplets typically will fall to the ground within a minute or two. So if someone coughs and then you walk past there 2 minutes later, it's unlikely that you're going to breathe in the virus. Aerosols are created by procedures that would create them. So dentists are really at risk. Those tiny drills that they use in your teeth will create aerosols. And then, so in the lab environment, they showed that the aerosol could last for 2 to 3 hours, but that's not taking into account that there's airflow, right? If you have your AC running or your heater in the outdoors, there's, naturally wind or a breeze. People might be moving and causing the viral particles to move around. And then, so to answer your specific question about the Diamond Princess cruise, so they found pieces of the genetic material, but it's unclear, just because you find the genetic material, whether that is actually infectious. So you need that whole sac to be there. And sometimes you can find viral RNA, which is usually how we test for these viruses, when it's outside of the protective sac that's actually infectious. So
The
I think
Rotary
it still
meeting.
remains to see if those particles are infectious or not.
No. Can I see you?
So,
No. My
Sita,
camera.
if somebody has a question, there's a raise your hand button. Otherwise, I can, Glenn, if you want to
Yes.
ask a question, go ahead.
Yes, thank you very much. That was a wonderful presentation, Parinaz. Really appreciate everything that you're doing and sharing it with us. So you have the power of several volunteer organizations here paying very close attention to what you're doing and saying. And together we have the capacity to raise certainly hundreds of thousands of dollars, if not more. So I would ask you, if today you could mobilize 500 volunteers, and let's just say $100,000 or $1 million, to stem the death rate and the infection rate of COVID-19, how would you invest the effort of those volunteers or that money?
Yeah, so I mean, I think there's two big things that right now we desperately need. One is tests, right? We need to ramp up the amount of tests that we're able to run. And so UCSF, the Mark Zuckerberg and Priscilla Chan have started
this
this
is our meeting.
scientific organization called,
This is a Rotary meeting? Yes.
I don't know who SETAD is, but if you could go on mute, that would be great. But basically they have set up their own CLIA testing lab at the BioHub, which is next door to the Gladstone in Mission Bay in San Francisco. And they have volunteers. One of my lab mates goes there every day and runs one of the robots to be running tests. And so what we desperately need is more tests so that we know how many people are infected. We can isolate the people who are infected and maybe asymptomatic. That would be fantastic. The other thing that we desperately need, again, is just beds, ventilators, triaging people. Part of the problem with 500 volunteers is that we don't have enough masks and things to keep you safe. And so we don't necessarily want to be putting people out in harm's way. But if we could be having people helping run tests, triage, kind of organize the response and get manufacturing or get tests out there, that would be really helpful, I think, in stemming things.
So we have a question from Parveen. Do you want to go unmute and ask?
Yes. So Patty Naz, you did a great job.
Thank you.
Obviously, I can say her name next. But I was wondering, this was really excellent. And I was wondering if you could do this same presentation. I know you have probably very little time to do this, but our school district is working really hard to support our students. I think that the more people know about this, the better job we can do to support our families. So if we could, if there's a possibility to have this repeated for our leadership team, our principals and science teachers who are working with kids and families, that would be great. And you don't have to respond now, but you can just let Gary know to let me know and we can get connected. I was telling you, I can make you some Persian food too.
And
Thank you.
Kathy Coyle, go ahead and unmute.
Oh, hi. Thank you, along with everybody else. It's just been fantastic. Before I ask my question, I would think that there's probably a couple people that recorded this today, and that should be made available to other Rotary clubs and organizations. But my question was, I was very intrigued by the iPhone self-swab test, and I had two questions. One is, do you think that you have a chance of getting false positives from that because people don't want to go too far up in their nose? And then how soon would you think something like that would be available, or are we looking out some distance? Thank you very much.
No problem. So I would say two things. So probably the FDA has been not, I think for the most part, not super excited about the idea of self-tests at home at the moment, although in the state of Washington, the Bill and Melinda Gates Foundation has been mailing people tests, which is basically those nasal swabs that I sent you or showed you guys. And they've been having people send back the swabs and then they run it in the clinical lab the way it usually takes a few days. And so I think it is possible to do those swabs at home, but definitely the efficacy of the test is based on how good your sample is. So I think that's definitely a problem. For us, probably it makes the most sense to have this at a point of care. So meaning a nurse or somebody in your doctor's office would quickly be able to take the nose, or even a drive-through, which is what a lot of places, South Korea did. I think some places in California are doing drive-through testing actually, where you'll just stay in your car, they'll stick the swab in, and you'll drive off. And so that might be more efficacious in this case. And then in terms of how long do we have to getting this deployed, my hope is now, again, because there's such a demand for tests and they really bungled it originally, they have been much less restrictive than they normally would be. And, you know, to be fair, usually you want your tests to be really reliable and you want them to be very well controlled, and you don't want just anyone to be able to run a diagnostic test. And that's why we trust our tests from our doctors. But you know, I think desperate times call for desperate measures. So my hope is, you know, within a year, if we really get mobilized, and we've been reaching out to lots of different foundations and companies to get us additional support and funding so that we can really accelerate this on a timescale that's much faster than is usual for academia.
Ashley, go ahead and ask your question. Thank you, Kath, for the question.
Yeah, I think if you are available, you know, I think this information to our business owners through the chamber would be a fantastic thing.
I believe they're recording this, so theoretically, I think the link could be shared,
Yes.
but I'm not sure.
So we are recording.
This
I
is for
think I
Rotarians.
chopped off the first part of the meeting accidentally, but we probably have a good 45, 50 minutes of this. And as soon as I clean up the link, I'll bounce it out to people. Todd, if you want to take it.
Sure. Bruce had asked a question too on how in the world South Korea got those tests out so fast.
You know, I think they just organized. It's not, the test is not hard. Every undergrad, most undergrads who have ever worked in a lab have probably run this test. It's a very basic test that I've run thousands of these tests in my, you know, research career. And so the test is not difficult. It's very shocking to me, again, that the original reagents designed by the CDC were just not very good. They were not very sensitive or specific, at least in my hands and in the hands of others. And it's, you know, if we had just used, you know, another country's test, which is published on the World Health Organization website, we could have, you know, gotten things running faster. And so I think there was an initial lag that really led us to be behind. And then, you know, South Korea mobilized, you know, they centralized kind of having the swabs, having the reagents, and they just mobilized everybody to dedicate themselves to doing that drive-through testing and testing everybody. We have not had such a mobilization, and we, you know, now have a shortage of even the nasal swabs to do the tests. And so on every step, you know, we're really struggling to keep up with what would be a very high demand for testing. So you know, basic competence and organization could have prevented this.
And Bruce had one other question too. If we don't have it now, why would we not be exposed the next time we go out and isolation would start all over again?
Yeah, so I mean, my hope is that they don't lift social isolation and social distancing until we are close to 0 cases, and until we are we are able to get lots and lots of people tested. Because, right, even if people were actually following the social distancing rules, which I just heard that Gavin Newsom shut down all state parking lots at parks and beaches because people decided this weekend was the time to go get a tan outside, and were flooding the beaches and parks and not keeping their distance. You know, if we truly followed it, and there's really a 2-week incubation period, theoretically, you know, we could shut this down in a month or 2. But if people continue to kind of flout the rules and think that they're immune, or it won't be that bad if they get it, we'll have exactly that problem where eventually we'll lift it. People will still be infected and spreading it to one another. So it really has to be a combination of stringent social isolation and very expanded testing.
Okay. And then Gordon Jones had a question. Should we be pushing more contactless payment systems, e.g. Apple Pay, due to the amount of germs on credit cards?
I mean, I think that we're exposed to germs constantly in our lives. I think that anything that has less contact is good, but, you know, I've been studying infectious diseases for several years now, and I don't wipe down my phone that often. That's not, you know, for the most part, generally, You know, you're not going to die from, you know, some germs on your phone, even though they are probably quite germy, and we should all be wiping them down more regularly. I don't think, you know, that's going to be the main transfer mechanism of disease usually. But I mean, it doesn't hurt.
Okay. Probably have 2 minutes. Any final questions out there? All right.
Hi,
With
this is
seeing none.
Roland.
Oh, go ahead, Roland.
I had a question. We run the sanitary district here in Castro Valley. We still are providing services with both the shelter in place and the stay at home order. The services we provide are essential, and we're still out there working.
Yeah.
One of the things that I have not been able to answer for my crew is when they're out and they're interacting with wastewater, and you know, some of the things that we do actually aerate the water so it becomes aspirated. How big of a concern is that, and what do you think might be the measures to keep my employees safe?
That's a great question. I don't know if there have been any studies that would look at how the virus can survive in kind of liquids. One question would be, is the liquid first treated, or the wastewater first treated before these aerosolization procedures? Things like bleach and alcohol kill the virus pretty easily. So if it's pre-treated, then theoretically most of the virus should be killed. And so aerosolization wouldn't harm things. But if it's not pre-treated, and based off of kind of what little data that we have, you know, that's what those N95 masks that are in such short supply are really useful for. They block 95% of aerosols from getting into the mask, which is pretty good. And then, you know, I'm not sure about the logistics, but, you know, if they can keep a distance from kind of where the aerosolization is happening, but still be able to operate the machine, that would also be beneficial. Wearing a face shield, any kind of protective equipment is definitely better than nothing. I would say probably the chances are low because I doubt that the virus, you know, even if it was able to last in that liquid, it probably wouldn't last, you know, indefinitely. In the air, it only lasts an hour or two. On different surfaces, maybe a day or two. And so probably it's not, you know, if you don't have to aerosolize it immediately, that's also another way, just by waiting a couple days, you know, doing the aerosolization procedure and then taking in the next batch.
Any final questions? Gary?
No, I'm not going to ask a question. Are we ready to talk about next week?
Sure.
First of all, Paranaz, thank you. This is a terrific talk. We really enjoyed it a great deal. So,
Oh, no problem. Thank you for inviting me.
Tell Melanie you get an A for this for sure. We appreciate your. So today we learned a lot about the biology of coronavirus. Next week we have another aspect of the coronavirus pandemic. Our very own Carol Weichel will talk about the economic side of this and how to survive in a bear market. So that's what happens next week. Take it, Todd.
So,
Thank you,
Todd,
Gary. Jim?
Very quickly, it'll take me about 24 to 48 hours to get the link out on that one. And we'll go from there. So please join us. Please look for it. If you don't see it, email me, call me, and we'll go from there.
Thank you, Jim. Thank you again, everybody, for joining. I hope it was educational, informative, and stay healthy to all.
Love you guys. Give you a hug.
Bye.
Take care.
Bye.
Be healthy.
Thank you.
Thank you.
Thanks. Bye-bye.
Bye. Thank you.